Hold on, I'm gonna go live and get this set with YouTube
Okay.
volume down real quick, so I don't have the same mistake I made last week.
Yep. Go do what you need to do. What do you think, Nikki? Are you there? Oh, can't hear you.
I unmuted for a minute. I like the one with the daytime outside.
Okay.
That one.
There we go.
All right, we
Dinner.
YouTube, so I'm gonna have to what do you call it? Edit this before we put it up.
Okay. Go ahead. And Frankie, are we, well, she's thinking, I don't want to interrupt her. Is this on Facebook Live too?
It's gonna be right now. Just, yeah, it's gonna be on Facebook. I'm
So
gonna,
it's Facebook Live and YouTube Live and the Zoom. So there's lots of ways people can get on.
correct.
Awesome.
All right, let me share this guy right now. Put this on our Facebook page right now. So we can start, people will start watching and we can say, we're not live yet. Well, we are live, but start till 6. There's plenty of time to get popcorn. Oh, the other thing I've got to get set up is, okay, hold on. We're about to be live on Facebook. So much to do. I need to get up the video of Erica's video, which was really good, by the way. Okay. Okay, we're good on Facebook. Hold on, let me make sure I'm not going to have a volume problem on Facebook. Okay, I don't have any. Okay. And then Erica's video. And then we are set, ready to go. Okay, let me, uh-oh. Because I have to do it from the link. That scared me for a second.
And I'm changing the subject again, but I think I told you guys like a month ago when my cousin got diagnosed with cancer.
Yeah.
She died.
Oh, Peggy, I'm so sorry.
I know, 5 weeks later. We weren't super close, but it's sad because it's the first of the 9 cousins.
Oh, Peggy, I'm so sorry.
Really fast.
Oh, I'm so sorry.
She got pneumonia and didn't want a ventilator and told them she would not be put on a ventilator because she didn't want her daughter to have to choose when to take it off because her cancer was so far progressed that she would probably, if she recovered from the pneumonia, she'd be close to death from the cancer anyway. And she died that night that they told her she had pneumonia. That was fast.
I'm so sorry. I'm so sorry.
Thank you though. I know it's just scary.
Yeah.
And I'm on my 4th grandpa that I know of that died of COVID.
Oh my gosh.
Classmate today was the 4th one.
It's very rough. All right, we're 10 minutes out. I think we're all set. Quiz me. I've got the video ready for Erica. I have the PowerPoint ready. The only thing I have to remember is to do the sound check when I load. Everything's running, everything's live. So I think
I'm able to share. I've got my stuff on.
yes.
We have 2 attendees.
Hi everyone. We'll start right at 6. And so this is different. So Zoom webinar, you're in Zoom webinar is a little bit different from Zoom meetings. If you've never done a webinar before, you won't be able to see you or hear you, but you'll be able to see and hear us. Otherwise though, it works pretty much the same. There's a chat where you can send in questions. And then at the bottom, there's also a feature that's Q&A. So you can either put questions up in Q&A or you can put questions in the chat and we'll be monitoring that for you. Sorry, my granddaughter's crying. And I think that's it, right? We'll get going right at 6 o'clock. We're anticipating, we're hoping we have a good crowd. Yeah. We'll just see. Welcome, welcome. We see another person's added.
And Sarah.
There we go. Turn up the volume. Hold on.
Will this meeting expand to 7:30-ish?
Possibly. That's such a great question. One of the things that we were going to announce is this one might go over a little bit, but you're welcome to drop off whenever you want. The reason it's going to go over a little bit is one of our presenters is the fabulous Mrs. Hernandez. And we thought you guys might like to have a little bit of a sky show so that you know what stars are up and what to be looking for this week. So this one might go just a little bit past the intended time of 7. Will it go to 7:30? I don't know. We're also willing to stay on and ask questions or answer questions that you might have specific to the expo. So from 6 to 6:30, we have our one of our presenters, Erica Zotek, and then we have Miss Hernandez, and yeah. So we're hoping we're planned for an hour. We know we're going to go a little bit over because we figured you guys might like to see a little bit of a sky show, and so we might be a little bit over. Hopefully not to 7:30 though. But we'll see. We'll see. And we'll stay on and answer questions too.
Yep. It might depend on the amount of questions. It might just be questions that are going over and maybe a little bit of demonstrating some answers. So it just depends on the questions. Definitely want to show you the night sky tonight though. And I know here in our area, we just had a beautiful sunset and we should have a good night for going out and looking at the stars. So it'll be fresh in your mind. You can step right outside and hopefully see everything that I try to show you. Yep. All right. We're getting some numbers. Hey, welcome everyone. Good to see you. We'll get started in a few minutes.
Hi, welcome everyone. We're so glad you could join us. We're going to get started right at 6 o'clock. I am Deb McMullen. I am the coordinator of science for School District U46. And our plan is to end today normally at 7, but we might be a little bit late tonight with our ending. You can leave whenever works best for you. But we might go a little bit over because we're kind of thinking about we're going to be showing you some night sky. So we might go a little bit over this evening.
There was a question about Are we going to create a project for STEM? And I shared with them that we will talk about what you can do for STEM Expo and give you some ideas.
Yeah, that's the purpose of the STEM Expo kickoffs, is to give you guys some ideas and also to introduce you to some science. And so we'll kind of get into that a little bit in a moment. But the idea is to teach you a little bit about the 7 concepts, the 7 cross-cutting concepts involved with science education, to also talk a little bit about the fact you're going to be using some scientific practices, science and engineering practices, and then also give you guys some ideas and introduce you to some local scientists. So we hope that you enjoy the evening.
And we'll give you a little overview on how the STEM Expo is going to work this year, since it's not going to be in person, but we've put a few things in place to make it just as fun as it's always been. And we're going to try to help you generate some ideas today, get the gears going up here, get the thinking going so that you get some ideas, a list of your own of things that you could start pursuing now in November to be totally ready in March when the STEM Expo actually happens.
Tonight, right now, it might be a good idea to start getting people hooked for next week, Ms. Hernandez, if you want to talk about our special guest for next week.
Yes, next week, I'm going to do a star show again with a little bit of a different connection than this week, but we also have a former student of mine. Some of you know I've been a teacher in the district for 30 years this year, and I taught at Eastview Middle School, and I had a student, and she loved the stars, and she loved the star party that I held, and she also loved dinosaurs. She really liked paleontology too. Well, it turns out she went to college for paleontology, and she's the collections manager at I think it's called the Salt Lake City Natural History Museum. She's basically the dinosaur collection organizer, and she does programs, and she's going to be here next week live. She grew up in Bartlett, and she went to Eastview and Bartlett High, and I think Centennial was her elementary school. And she does paleontology as a career now. She's done some really awesome digs out in the middle of nowhere in Utah with the little picks, and with a little team. They have to fly in on a helicopter because it's so out in the middle of nowhere. And they have found dinosaur bones, dinosaur fossils, fossils of bones. And she's going to do a little program next week. And then I'm going to do a little star program next week. So that'll be a fun event. Again, it'll be 6 to 7 o'clock, same time, same Zoom link if you're on Zoom next week, and it'll be great fun.
We're so excited. All right, well, it's exactly 6 o'clock. So if you have never participated in a Zoom webinar before, it's a little bit different than a Zoom meeting. You can see us and hear us. We cannot see or hear you, but you can still interact with us. And you would do that through chat, and you can do that through the Q&A. We'll be monitoring that. You can ask questions as you see fit. Now, our first presenter, I'm going to go through just a very quick overview of some things about how to set up today and why we are so excited to have all of you here. And then we have 2 presenters today. So our first presenter is going to be Erica Zotek. She is actually a professor at the Roosevelt University. So she actually sent us a video because she's teaching tonight, but she's going to get her students started in the lab, and then she's going to come in to answer questions after her video. So she submitted a video to get her kids started, and then she's going to come in and answer your questions about structure and function, one of the 7 cross-cutting concepts. And then our fabulous Peggy Hernandez is going to do a presentation about patterns with cross-cutting concepts. And the whole idea of this is to introduce you to the 7 cross-cutting concepts that all scientific phenomenon is embedded in. And the reason that we want to do this is we want to introduce you to a bunch of different ideas of things that you can potentially do for your project. We want to expand your horizons and get you thinking about different ways that you can be looking for science phenomenon that you can explain in your STEM project. We've also provided you with some other resources in the emails that I've sent. And so there's a bunch of resources in those, but you can always contact us too for more information if you need more help on that. So I'm going to screen share really quick. And before I do that, I'm going to make sure I share my sound. You see there the video of Miss Erica. Hold on a second.
Miss McMullen, did you know you're muted?
I'm going to be that person today. Thank you. Sorry. All right. So let's just frame what we want to do with you guys this afternoon or this evening. The first thing that we want to work on, why won't my slides go? Okay, we're going to introduce you to the 8 science and engineering practices, the 7 cross-cutting concepts that you'll be utilizing to do these projects. We're also going to give you some ideas featuring cross-cutting concepts of patterns and structure and function. We want you to set your calendar because the STEM Expo is a different time. Normally it's in February. We've moved it back to March 20th, 2021. So set your calendar. That will be the day that you will be providing us or presenting your project. So March 20th, 2021. Cross-cutting concepts are what all science falls in. All of our phenomenon falls within one of these. Patterns, cause and effect, scale, proportion, systems, energy and matter, structure and function, stability and change. And so we decided to organize our Wednesdays around these cross-cutting concepts to be able to give you clear-cut ideas of things in the community that you can be looking for in order to explain, just to give you some more ideas. These are the 8 science and engineering practices that you're going to be using in order to explore that cross-cutting concept. These are the scientific practices that we teach in School District U46. Plan and carry out investigation is the third one. And when we think about science, we very often think about, oh, I'm going to be in a lab, I'm going to be doing an experiment of some sort. Well, that's actually just one of the science and engineering practices. You have to be able to model. You have to be able to engage in argument. You have to be able to construct explanations and analyze data. And so in U46, even in this time of COVID, we are working on all of these science and engineering practices. Now, it might be harder to do number 3, plan and carrying out investigation, but that's okay because we're going to be working on fine-tuning the other 7. So we're always doing science and engineering in School District U46. Today, we're going to work specifically on structure and function and patterns. And so first you're going to hear from Erica Zotek, who is a Roosevelt University professor. She's going to speak for about 24 minutes in a video. We're going to be watching for her in the inbox, and then we'll bring her over so that you can ask her questions. And then when she's done, then Peggy will take over and she's going to explain to you patterns. All right, so let me exit out of this guy. And let me bring up, here is Miss Zotek.
Hi, I'm so excited to be here with you today to talk about as you get ready to prepare for U46 STEM kickoff events and you get ready to do your projects for the STEM Expo coming up in March. My name is Erica Zotek, and I am a lab manager. I am a lab manager at Roosevelt University, and what that means is that I'm responsible for getting all the supplies and everything ready for all of the science labs that we have on campus. I also help the instructors and the students who don't really know exactly how to do the experiment. I help them along so that they can go ahead and do the experiment correctly. A little bit about me is that I am a cell and molecular biologist. I actually went to school in I went to grade school in Chicago. This is my grade school up in here. It's called Norman Bridge. It's on the north side of Chicago. And then I went to high school at Glenbrook South High School in Glenview, Illinois. I didn't do I was I took a lot of science classes at the high school. I took over 5 years worth of science courses over there between summer school and taking double classes. It was a lot of fun. I really enjoyed it. They had some really wonderful labs then. And then I got my bachelor's degree at Quincy University. Quincy is a small college in Quincy, Illinois. Here it is right here. It's right on the Mississippi River. It's about as far west as you can go in Illinois. And then I came back and I went to Illinois Institute of Technology for my master's degree. That is on the south side of Chicago, right across the expressway from where the White Sox play baseball. So if you're familiar with that area, you kind of know where IIT is. So in school, I did one experiment, one science fair project, and that was to make a perfume atomizer. A perfume atomizer is basically a device. These are the old-fashioned kind, right, that have the bulbs on it, that helps push the perfume out of the bottle and out onto you or another surface. And that works by air being pumped through this. You press this little pump here and it pushes air across this way. And because of the air being forced across, it creates a negative pressure and it actually brings the perfume up and out this other tube. So that's how a perfume atomizer works. This is actually how a lot of our pump dispensers work nowadays. When you push down on that pump, it pushes the air out and it brings up the whatever material that is, whether it's like a cleaner, a disinfectant, perfume. It brings that out so that you can apply it to wherever you need to. So today, what we're going to do is we're going to talk a little bit about structure and function. Okay, so structure. What is structure? In its basic form, structure is what basically the shape or form of something, right? Your hands are a structure, right? The eyes, this is a specific structure. A nose is a specific structure. This tail on this little squirrel is a specific structure, as well our ears. And you can see that even like we see the little bit of ears here on the squirrel, and they're slightly different from the horses here. The eyes are different from the horse, to the squirrel, to the cat. You see the cat, these are positioned way in front on the squirrel, and the horse are positioned way on the side. And you can see some little differences too in the hands. These are human hands, right? And these are squirrel paws. They're a little bit different than human hands. And they all have very unique functions, right? And so what is a function? A function is literally what something does. So our hands, we can use to touch, to grab, to feel. Obviously, a squirrel has ones that are very similar. Horses have hooves, right? They're used for running and stomping on their enemies. Cats have paws, and those are for batting things. They can even catch things with their paws. So they all have specific things that they do. Now, plants also have specific shape and function, right? We have our plant here where we have petals and the flowers, we have the leaves, we have stems, we have roots. And some plants even have thorns, right? Unlike a horse who can run away if it gets scared or if a big animal is going to attack it, a plant can't run away because its roots keep it where it's at. So this is why a plant may have some thorns to go ahead and function as defense, where a horse can run, a squirrel can run. Now let's talk just a quick little minute about the eyes that are different here on our cat and our squirrel and our horse that we mentioned. If you notice, the cat's eyes are way in front so they can see everything in front of it, right? Where the squirrels and the horses are on the side so they can see more around, more not only in front, but on the side and a little bit behind them. That's because a squirrel is a predator. So its eyes function to look for things in front so it can catch and chase, right? Where a squirrel and a horse may have to run from a predator like a cat. So they have to be able to see all around them. So this is why their eyes function a little bit differently because of where they're located. So we have the big anatomy that we see, right? Everything that we see, our hands, our eyes, our noses. But we also have a lot of structure beneath what we see just when we meet somebody or see an animal. Our brain has a specific structure, right? It's got these bumps and everything in it. Our heart has a specific structure. Our lungs have specific structure. They are all different because they have different functions. A brain needs to communicate. And if you can see here, this is a mammalian brain like a human brain. So cats, horses, dogs, their brain is shaped similar. This is a bird brain. It's a little bit different. And this is a fish brain. And again, that's different because they all function differently. They're all doing different things. This is a heart again. The mammalian heart is similar in all mammals, so it's very similar in a horse, a dog, a cat, an elephant. Much bigger in an elephant, obviously, but they all look about the same. These are lungs, right? So our lungs help bring oxygen in and carbon dioxide out, right? So we have to bring in our air in order to breathe, but we also get rid of any of the waste, which is a carbon dioxide, through breathing out. So these are human lungs. These are pictures here of different types of lungs. You have like a salamander lung, you have a frog lung. This here is like a reptile lung, a rabbit, which is a mammal, so it looks very much like ours. And this is a chicken lung. That one's very, very different, but they all function very similarly, but they do have some differences because of how some of these animals breathe, right? And a frog can sometimes breathe underwater, or they can hold their breath really long underwater. We can't do it for as long. So their lungs are a little bit different than ours. So they all have specific functions, right? Our heart is to pump blood, so we can get the oxygen from the lungs into the blood all over our body. Our brain basically tells us how to move our muscles, when to run, when to jump, when to laugh, when to cry. So if we look even further, we can see that this is the brain. Now, these are actually brain cells, okay? These are all stained with different colors so we can see them. These blue dots are what are called nuclei, right? We'll talk a little bit about that in just a moment. But as you can see, there's different types of cells. That's why we have these yellow ones, and there's different red ones here. These yellow ones have long tendrils that reach very far. So some of the cells in your brain start in your brain, and they reach all the way down to the bottom of your foot, because those where the nerves are. So this is where we can see some of these cells in here. So we got different types of cells. You got astrocytes, you got ganglia, a lot of different types of cells. This here is a blow-up of what a heart cell looks like, or many heart cells. Again, these are stained so we can see them. The nuclei are the dark pink, and these are long, skinny, like strings next to each other, are heart cells, because it's a type of muscle, right? So it has to pump, it has to be strong, it has to look like muscle, and it kind of does. These here are our lung cells, and our lung cells are they look like they're kind of holy, like they've got a lot of holes. Well, because our lungs take in a lot of air and oxygen that way, we have to, it's like a big sponge to absorb all of that air. So this is why they look like that. So they all have different functions, right? Because we're taking the air in, we're breathing air out that we no longer need, the carbon dioxide, and it's holy, so it can absorb all of that. These are structured, our heart cells are structured differently. They look like muscle because it is a muscle, but they look like what we think muscle looks like because it has to pump, and it's pumping all the time. It's always working, right? So it's nice and fit. And our brain cells, they have to go very far, some of them. So that's why they have these long tendrils, and they like to communicate with each other. They have to communicate, right? If they don't communicate, then you don't know to pull your hand away if you're touching something very hot. So the way they're structured depends on their function and how they work. So here's some plants that we can talk about. Now, most of us have eaten carrots, and we've all sliced through a carrot, and we see all these rings, right? So this ring here is what's called the xylem. The xylem are these holes here, and what those holes, they're big holes because they function to pull up the water that a plant needs. So the stem, the carrot is kind of, the carrot grows underground, so it has to pull up a lot of the water. There are little roots that come off of a carrot, but it has to bring that water up to the leaves so the carrot can actually grow. So this is how it pulls that up. And what's really neat too is if you were to take a slice of a branch or a leaf and look under the microscope with that as well, you would see these same type of xylem cells, because that's how the water gets all over the plant's structure, right? Just like we have blood cells and where we need them, instead of blood cells, the plants have xylem. And then what they also have is called phloem. That's what these cells are here, this little bunch here. And the phloem on a carrot is right here. And what that allows when the plant makes food from the sun to get it to the root so the root can grow. Like here's an example of a plant growing underground with its roots, right? So we take the plant grabs energy from the sun and makes food up in the leaves, but it has to bring that food down to the roots so the roots can grow. And that's what this picture is. This is a picture of roots that are growing. This is undergoing what's called mitosis. So it's making more cells. Like here is one cell getting ready to divide, right? We have two nuclei, and this is one cell what used to be one cell is now two cells. And here they're pushing apart in order to make more cells so that root tip can keep growing. So they do have different structure because they're doing different things, right? In our stems and in our parts of our leaves, like the vein part of our leaves, we have our xylem and phloem, the xylem to bring up the water, the phloem to bring down the nutrients to the other parts of the plant. And then in the roots, they're growing. So the cells are very different there. Now, what's also really neat is that plant and animal cells are both very similar in many of the parts that they have. Now, this is just a cartoon drawing of an animal cell, right? Some of the main things that we need to care about in our cells that are both plant and animal cells is this membrane. This membrane here keeps the things in the cell that need to be in the cell, like our nucleus and our mitochondria and our vesicles and everything else, and our Golgi bodies. It keeps everything in, but it keeps other things out that we don't want to get into the cell. This membrane, and both in plants and animals, is made up of some fats and some protein. So this is kind of why we need to eat a little bit of fat, because our cells need that in order to stay protected and healthy. But they also have what's called nuclei. A nuclei is where all of our DNA is. The DNA is basically the code for what makes you look like you and how you respond to things and how you look and how, all that different things. And what's really, really neat is the same species. So humans pretty much have all the same DNA, but in different people, certain parts of it are turned on and certain parts of it are turned off. That's why somebody may have blue eyes, somebody may have brown eyes, somebody may have curly hair or straight hair. That's all in your DNA. And same thing for a plant cell. Here's a nuclei, right? Here's the cell membrane. And the plant, as we saw in here, this darker spot here, this is actually a cell wall. So the plant cells actually have an additional little outside protection, but they all have a membrane that keeps everything in that's supposed to be in there. So they all have specific structures. They all have specific functions. What helps make the plant green are these chloroplasts. These are these green parts here. This is where the energy is taken from the sun and made into food. So everything has its own structure and its own function. So I know this is a lot of information. It's actually a lot of really cool pictures, and we can kind of see how we go from the big pictures, right? From why we have structure and function with our hands to touch, to grab, with our eyes to see and process information. With tails are great for balance when animals jump and when they leap, and they're also good too if you're an animal and you understand the communication, how to talk to each other. Our ears are for listening and how to pick up things. These horses have big ears, and they kind of turn around so they can hear the predators coming up. And plants, they have their structure, right? They have the stems that go up and the leaves that collect the energy from the sun in order to make food, not only for themselves, but sometimes for us too, right? And the fruits and vegetables that we eat. And they have their roots that pull up nutrients from the ground, so they can make everything, and they can protect themselves with thorns. And then we see how we can bring that a little bit more specific. And we look at the brain, how it has its structure and function, and the heart and the lungs. And even though it may be a little bit different looking in other organisms, they all have the same type of function. And they're very similar because the structure is very similar because the function is very similar as well. And then we can look at the cells, and these cells are different because they're doing different functions, right? We have the long cells in the brain, and the long nerve cells and ganglia and everything that have to be long so that you can feel something in your foot and it registers in your brain really fast. And your heart cells have to be kind of strong and long in order for it to pump all the time. You need a little bit of air in your lung cells so that you can pick up the oxygen from the air and get it processed and put it all over your body. And even in plants have the same type of thing, right? They have our xylem to bring up the water from the ground that we can actually see this in the carrot. And that's why carrots are pretty neat, that we can see this happening. Or where it's happened. You have the phloem, so the nutrients get down, and so our root tips can grow, so we can get bigger plants and more plants. And our cells have these membranes, they have nuclei. Not all cells have all this other stuff like the mitochondria and other things. Like bacteria are considered cells, but they don't have everything that an animal cell does. They have a few things that are different and a few things that are the same. our plant cells have a lot of this, the same stuff that the animal cell does too, but it also has a cell wall because our plants are a little bit more structured, right? Because they can't defend themselves, so they have to have cell walls in order to help with that. So how can we go ahead and look at this? So we don't have microscopes at home, but we can do a couple different experiments. And there, you may have done something similar to this, but I like this one a little different. Most of us have at one time or another put celery in a colored water dish, right? The dish of colored water like they have here. And we see the xylem working because the tips of these celery stalks are different colors depending on the color of the water that they're in. But we can also do is see what different nutrients do, right? So instead of just doing colors, we can put in different nutrients. We can do plain water. We can do Gatorade, which has a lot of phosphates in it, so that we can go ahead and see what that happens in salt and sugar. And then if you want, don't necessarily have to, but I think it'd be really cool. Like if you use red Gatorade, so you have plain water, but you also have like red food colored water to do the same thing. So you would do very similar to this. You'd have one with water, one with food coloring, one with Gatorade, one with salt, and one with sugar. And you put your celery in there. Now you can optionally cover the lids with some plastic wrap like saran wrap so it doesn't evaporate real fast, or if it doesn't knock it over, it doesn't spill really bad, especially if you have food coloring because you don't want to stain anything in the house. But you can see what happens to these leaves, like after 1 day, 3 days, 5 days, 7 days. See if there's something happening different with the leaves, with the Gatorade, the salt, and the sugar. And that's evidence to see the xylem functioning properly, right? Pulling up, but also to see how it affects the nutrient, how the nutrients affect the plant. So we can see some of that function too with the phloem. Another one that's really neat that you can do is you can actually take a basil plant or grow some basil plants. So you would need some potting soil, some small containers, which could even be plastic like cups that you have at home. A ruler, again, water, food coloring, Gatorade, salt, and sugar. Do it basically almost like we did in the celery experiment, but instead of watering the plant with just water, you can water a Gatorade, salt, and sugar, and you can also see what happens to the leaves. Do the leaves change color? Do they get smaller? And this is why we need a ruler, because what you would do in this experiment is you would set up your soil, your pots with soil, and then you'd push one basil seed in each plant the same distance. So if you go to the first knuckle of your finger, so that way you know how deep it is in each and every one. And then you let the plants, water them with regular water, let them grow until they're maybe about 2 inches tall, which isn't real big, but 2 inches tall. So they all start at the same point. And then once they start at 2 inches tall, then you can start watering them with your different conditions, right? You still want one with water, one with maybe just food coloring, and then one with Gatorade, salt, and sugar. And you do that for a few weeks. It's a little bit longer of an experiment, but it's kind of neat. So you can actually see what happens. And then after a few weeks, you can see which one's taller, which one has bigger leaves, which one may have leaves that are wilted or brown. Make all that information, write all that information down. Take pictures, right? So this is how we can see the evidence of the structure and the function. The xylem will be obviously pulling up the water from the soil. Hopefully, even when you use the food coloring, you might even get a little bit of coloring in the leaves of that basil. But you'll also see the functioning going on too, because if there's too much salt or too much sugar in the soil, the plant may not be able to function properly and may not be able to grow properly. So that's how we can indirectly see something if it works without using a microscope. Another one that's kind of cool is bird beaks, right? Birds all have different style beaks as shown in this drawing here. And so they eat differently. So what you can do is take aluminum pans, slightly bigger ones, or you can use slightly bigger Tupperware containers and put some moist soil in one, and then cut up some pipe cleaners and put that in the soil. And then in another one that doesn't have any soil, just dump a bunch of seeds and grains in there, like pumpkin seeds, sesame seeds, rice, barley, quinoa, anything. Just mix that up and throw that in another dish, in another container. And what you'll have a forceps, which are also called tweezers, and pliers. And what you'll do is have an empty jar or cup on the side and a timer, and set your timer for 15 seconds. And what you'll do is you'll do one round with your forceps, your tweezers, and pull out the worms out of the soil one, and then another 15 seconds and use the pliers and pull out the worms out of the soil one and put those in your empty jar or cup so you can count them. And then do the same thing again with your seeds. With the seeds, you have to actually crush them, not just pick them up, but you have to pick them up and crush them in your grains too, and then put them in your cup and see how many you have with that after 15 seconds. So I'll go ahead and show you some of this and that we can do. So this is a basil plant, right? So what you would do is you could, like I said, you could put your soil in, and then like your empty soil, and you could push your basil seed down, like just until a certain part of your finger, but in the same spot every point in your finger. So that way the seeds are at the same point. Now this one's a little taller. We've let this one go a little bit taller, but you would measure it. You can measure it on the edge here or in the soil. Sometimes it's better to stay on the edge because sometimes the soil gets knocked down. So once you get to maybe 2 inches, which would be about here on this plant, here, what you would do once all your plants are lined up and they're all that same height, then one would get plain water, one would get Gatorade, one would get sugar water, one would get salt water. Now, when you make up your sugar water and your salt water, you want to make it all the same, right? So maybe you do 1 liter of water and 2 tablespoons of sugar, and then 1 liter of water and 2 tablespoons of salt. Or you can increase it or decrease it depending on how quickly you want things to happen. But you might change, you might get some color, a different coloration in the leaves. But you do want to make sure though that you put everything in the same, like next to each other in the same window or on the same shelf, so they all get about the same amount of sunlight and everything else too. Okay, so this is, and obviously the basil smells really, really good. For the bird beak, so this here's a little dish with some moist soil, and there are some, can't really see them too well, but there are some white little, here's one that's not in there, white little pipe cleaners in there. So you would take forceps or tweezers, right? And you could go in here and you pull out your worm. I pull out a worm and you put that to the side, and you would do that for 15 seconds. And then you would take pliers and do that for 15 seconds. Oop, and that's harder to do sometimes because there's other parts of the soil in there. Because different birds have different types of beaks. And this is a perfect example of structure and function, right? Because these beaks function differently. This is a different type of a beak than this. So their structure's a little bit different because they function a little bit differently, how they grasp and everything. So here I have a little container of some seeds. I've got, it's hard to see here. I've got some rice and some barley and some pumpkin seeds in here, and you just dump it in, and you would do the same thing. But now what you have to do is when you pick up something, you not only have to pick it up, but you have to crush it. And some things you might not be able to crush. Oop. Or it might go flying across the room, right? You can also pick up some bird seed at the store. That's really quick too. If I use a forcep, or not a forcep, a plier, and I pick up a pumpkin seed, I can crush that. So that would be one you put in your cup to count. If you don't crush them, you can't count that, because that's what the bird needs to do in order to eat, is to crush that seed and eat it. So those are some experiments you can do to see structure and function. So I hope you've got some good ideas. I hope you're able to get some information from me today. I cannot wait to see what some of your experiments are going to be. I'm really excited to help out with this. It's always so much fun. And you know what? It's a great way for you to learn something new, or to just get more knowledge on something you already have. So thank you so much for spending some time with me, and I can't wait to see you soon. Bye.
And so now we have her live. Miss Zotek, how are you? Do you want to turn on your, how are you?
Good. My
okay?
camera's not turning on. I don't know if you have to do that or
well,
not.
it's okay. We have your fabulous picture. I was wondering if anybody has any questions for Miss Zotek, either about what she talked about with structure and function, or if anybody has any questions about any of the experiments that she mentioned. I do see somebody's raised their hands. So Chris, do you have a question? Do you want to put it in the chat or the Q&A? Sometimes that, all right, so we have,
Ria has one.
all right, go ahead.
Ria asks, can one thing have more than one function?
Oh my gosh, great question.
So yeah, you know, our mouths have more than one function, right? We can breathe through our mouth if we're running, or if our nose is plugged up. We can talk through our mouth. We eat through our mouth. So yeah, certain things do have more than one function. Likewise, some things only have one specific function. So it all depends on what needs to be done and how it's adapted.
That's a great question. Does anybody else have any other questions? Oh, so one of the other things that you should know is that during the STEM Expo, it's not a judged event per se, but you will have knowledgeable people who will be listening to your presentation and asking you questions. And Ms. Sotek will be one of those people. She's done it for many, many years, and she often brings some of her students to help out as well. So she might be one of the people that's asking you some questions. I see that there's a couple more questions that have come in. Do some things have functions that aren't necessary? What a great question.
Actually, there are a few things there are some organs and other things that we have that are not 100% necessary, right? We used to have, some people still have this organ, it's called a gallbladder. We really don't need that anymore. Or our appendix really too. Our appendix used to be much bigger. It used to have a function to filter things, and now we don't use it anymore. And that's why some people have to have surgery to get rid of it. Most things do have some function. It may not be very necessary. Well, I think the best way to put it, it may not be always necessary, right? There's some things that are used only maybe a few times in their lifespan. However, most larger organisms, and especially really tiny single cell organisms like amoebas, pretty much everything they have does need a function. Again, like I said, in humans, the biggest one that I can think up off the top of my head is the appendix. We really don't need that anymore, but we still have it. So it's not necessary anymore to have a appendix, and this is why people have surgery to get rid of it if it becomes inflamed. And we're able to filter things through like our liver and kidneys instead of through our appendix now. I hope that helps answer the question.
It was a good one. There was somebody wanted clarification on when you were talking about the water, the sugar and the salt. Could you just clarify that? It looked like they were maybe interested in the experiment, but weren't quite sure what you meant about the sugar, the water, and the salt.
Yeah, the water would be our control, right? Because most of us just water our plants with plain water for them to grow, especially if plants are growing outside. Like if you have a vegetable or herb garden, or the farmer's crops are typically just water is sufficient for them to grow. And then if you have another plant that has sugar, just sugar, and then you have another plant that has salt in that water, right? Because you can't just put dry salt or dry sugar in the soil. You have to mix it in the water in order to water the plants that way with either sugar or salt, to see if there's a difference in how that affects the plants. Because I hope that clarifies that question.
Yeah, I think that was good. And then one other question, can plants have more than one function?
Oh, that's a great question. Yes, plants can definitely have more than one function, right? A lot of our trees, they can provide us shade. They provide us with oxygen. Some of our trees will also provide us with food, right? If you have an apple tree or a pear tree, we can get food from that. Even smaller plants can provide more than one function. Soy is a great example of this. Soy we can use as food, right? Edamame, soy sauce, all that we can use as food. Soy can also be used as, we can take the actual plant structure, not the seeds, but the plant structure, and you can make rope out of that and various other products. George Washington Carver did some wonderful teaching of some of the sharecroppers on how you could use soy and corn like 50 different ways. So that way the sharecroppers could get as much use out of the plant as possible. Obviously corn, we use corn to eat, we use corn to feed our animals, but a lot of the stalks on corn are also used to make cellulose fibers. So a lot of like filters, or like a lot of some of the, what are they? Even our disinfectant wipes, those are made out of cellulose fibers, and that is actually plant product. Those are the plant stalks primarily from like corn and soy. So yeah, our plants definitely have more than one function.
All right. I think that no other questions have come in. We really, really, really appreciate you very, very much. There's another question about animal cells and vacuoles like plants. And how about this? That might be a really good experiment for you, or a good piece of research for you to do, to look and see about vacuoles versus animal cells and plant cells. So we really appreciate you. We thank you so much for taking your time. I know you took some time away from your students to be here with us this afternoon or this evening. And we really look forward to seeing you at the STEM Expo.
Well, thank you so much. I appreciate helping with this. And it's always wonderful to see the great projects the students come up with.
Thank you.
Thank you.
All right, Mrs. Hernandez, you are up.
All right. So I'm going to share the screen. And get to my program and start with the phenomenon, because that is it. That's where we always start when we do any kind of a science investigation, or if you want to call it an experiment, whatever you want to call it. We always start with something that's interesting, a phenomenon, an amazing sight, a surprise, a discrepant event. That means something that you saw happen that you totally didn't expect to happen, and it's not magic. What is it then? What happened? Anything that interests you or surprises you is called a phenomenon. And that's usually where we start science. Now, sometimes it's a problem too. It can be a problem, something that you want to find a solution to. But either way, that's what you need to start with when you do your Science Expo project. For example, I don't know about you, but in the past week, I have certainly noticed that I cannot work out in the yard after supper. Now, for some of you, if you're students, you really couldn't go outside and play after supper this week, even though it was super nice outside, like plenty warm. You could wear a t-shirt or even shorts, but it was dark by 5:30. And that is not how it is in the summer. So what is up with that? Why does this happen every year? Like, I've noticed it's dark earlier. And I also have noticed in the springtime that it starts to get dark later. That's a phenomenon. A phenomenon. That's it. That's where you start. Like, it might even be a problem. Like, can I change this? Can I make it be light later? Because I want to go out inside and play after supper. But this phenomenon of it getting dark earlier is something that's, well, it's exaggerated by the human time change that we do. That's something that us humans decide to do. We spring forward and fall back. Even if we ignore that, it's still getting darker earlier. So what's the next step then? All right, so I want to do a science project on it getting dark earlier. What do you do next? Well, you think about it. Come up with a list of questions, gather some information, talk to your grandparents, talk to your brother, talk to your mom or dad, talk to somebody about this getting darker earlier thing. Listen to what they say. Do you agree? Is there an investigation you could do? Are there things you could check out? Doing this questioning part leads to the next steps in your project. You're already doing a project if you do this part. So let's do some observing. Let's say we want to do an observational investigation. That could be a STEM project if you do a very careful observational investigation. So let me go to a different screen, and I even want to go like this so we can see the whole thing. There we go. All right, so here it is on May 11th, 2020. So we're talking months ago. So back in May at 6:41 in the evening, right down here on the bottom, there's the time, 6:41 in the evening. The sun is just about to set in the west. I'm going to fast forward time a little bit so we can do a little observational investigation. I want to wait until it gets dark. When it gets dark, I start to see darkness sort of enveloping the screen. We're not quite at sunset yet. Slow it down right there. There we go. we start to be able to see Venus. I see the sky turning gently darker. It's 7:49 right now. I would say it's probably close to 8 o'clock when the sun set on May 11th. But hold on, I can change it. I'm going to go back a couple of hours. So instead of it being at 8 o'clock, we're back at 6 o'clock. But let's go to June 11th. Are you ready? I'm going to fast forward an entire month at a time. Instead of being May 11th, now it's June 11th at 6 o'clock. Let's fast forward time and see what time it gets dark now. So it's 6 o'clock. Watch the clock on the bottom. I'll fast forward a little bit. It's starting to get noticeably dark. A little bit after 8 o'clock. A little bit after 8 o'clock, we got 8:16 here. I forgot what our first one was. And I'm just estimating here. I don't have exact numbers. I'm just observing and seeing what I can notice. But it's a little bit later than the May one when we can spot Venus. Maybe that'll be our teller of how dark it is. When we can see Venus in the evening sky, we know that it's pretty much dark. And whoa, it's 8:38 and I still don't see any stars. It's almost 9 o'clock and I can still see light on the horizon. And that must mean that that the sunset is getting later and later. So let's go forward again. I'm going to go backwards 2 hours again. Now let's go to July. Here's July at 7 o'clock. Let's fast forward time. When does it get dark in July? About 8:20. About 8:20 in July is when it started getting dark out. Let's hold on. Let's go. I'm going to go back another hour. Let's go to August 11th. And August 11th at 6:20. Whoa, the sun is still way up high. I can fit a finger between the sun and the horizon there. On August 11th, 7:30. Ooh, 7:47. It's starting to get dark out. Before 8 o'clock now. That seems a little bit earlier. Hold on. Let's go another month. So I'm going to go back to 6 o'clock. Let's go to September 11th. On September 11th this year, at 7 o'clock, it was already getting dark out. By 7 o'clock, it's starting to get dark out. So wait a minute. Hold on. Let's go to October. I'm going to go back 2 hours. And we're going to go to October 11th. Hey, did you notice anything else about the sun, where it's setting each night as we go through these months? Not just the time, but where it is. Let's look at the time. Oh, I'm all the way back to 5 o'clock, 5:30. Whoa, 6:07, and it's starting to get dark out. And if I go back 2 hours and we go to November 11th, 4:08, 10 after 4, and it's starting to get dark out. That's where we are today. What do you think it'll be like in December? Is there a pattern going on here? A pattern? That's the cross-cutting concept we're talking about today. There's patterns in plants, in biology and cellular biology. There's patterns in science too. Let's see what happens in December. I'm going to go back an hour. Well, let's go to 2:47. And we'll go to December 11th. You ready? On December 11th, that's where the sun's going to be at 2:47, not even 3 o'clock in the afternoon. I'm going to fast forward until it gets dark. What's your guess? A prediction is an educated guess. If there's a pattern going on, your prediction will probably be true if you're following the pattern correctly. So here it is, 3:20, 3:30. Oh boy, 4:08, and it's already getting dark out. 4:08, and it's getting dark out. Well, what do you think it will look like in January at 4:08? Let's do that. I'm going to go forward to January. Here's January at 4 o'clock. Hey, let's look at February. Here's February at 4 o'clock. Here's March. Here's April. Look on May 11th, 2021, we're going to have a new moon. That's the moon by the sun there. And at 4:08, the sun is way up high in the sky still. What about June? The sun's all the way up here in June at 4 o'clock in the afternoon. It won't get dark in June until, 8 o'clock or so. It's already late again. So I'm detecting that there is some kind of a pattern here, and I can show you one more. I'm going to go back to those months for just a second. And we need to go to about 4 o'clock. I'm going to go through a whole year. Only it would be better to be at noon. 12 noon facing south. You know, the sun rose up in the east, it crosses the sky, and it sets in the west. Noon, that's when the sun is the highest in the sky. On June 11th, that's where it is. Let's see where it is on July 11th. Are you ready? Here we go. Here's June 11th, July 11th, August, September, October, November, big jump because the time change, December, January, February, March, April, May, June, July. I'm going to go faster. See if you notice a pattern. Pattern again, pattern. Patterns all over the place in astronomy. Here we are, July, August, September, October, November, December, January, February, March, April, May, June, July, August, September, October, November, December. Could you do an observational investigation and mark where the sun is in the sky every day at noon, or maybe once a month at noon for a year and find a pattern? Of course you could. Now, the thing is, once you find a pattern in the sky, it often makes you curious. Yeah, because you say, well, what's up with that? It leads to more questions. That's what science is all about. So now I want to go to this next slide. So
May I interrupt you? 'Cause
yes,
we just made an amazing observation.
so go ahead.
Said that they feel like what you just showed look like a figure eight.
It is.
I think that's a thing that we could collect some
Yeah.
evidence around. I think that's a great phenomenon to explain.
It is. It's a really interesting phenomenon, and people have noticed that figure eight diagram for hundreds and hundreds since people have been alive. They realize that the sun is in a slightly different spot every day. Now, if you want to go into the why, well, that's an amazing phenomenon in and of itself. First, you got to do the observations to figure out that there's a pattern, but then figuring out why there is a pattern, you got to take your questions and sort of classify them. Like, what's the exact question I want to answer? A question that's not too big and not too small. The best questions always are the ones that ask for, that need a longer answer, that need an explanation rather than just a one-word answer. And if you can classify all the questions you have about that, sort of that figure eight pattern, and I said I wouldn't give away any answers. It's really hard to teach without giving any answers. It's called an analemma. If you ever want to look it up, it's called an analemma. And I'll bet you've seen an analemma somewhere before. You might not have known what it was or why it was there, but I'm not going to tell you where. So look up analemma when we're done here and see if you can figure out where you've seen an analemma before, that figure eight. It's not exactly a figure eight. It's sort of stretched out, but yeah, it's called the analemma. But these things that you're thinking, these questions that are coming up based on these observations can lead us to predictions. Patterns allow us to do that. That's why people thousands of years ago eventually figured out when we were going to have lunar and solar eclipses, because they happen in a pattern. You can figure out the pattern without even understanding why the pattern happens. So a project for the STEM Expo doesn't have to be mind-blowing, brand new information that the world has never heard of. It can simply be looking for a pattern and finding a reliable pattern and being able to predict. And we definitely can predict if we find patterns. One thing I thought of was I was just kind of eyeballing it when we had sunset, like when it kind of got dark. Wasn't super accurate. I probably could gather some data, and I did. This took me a few minutes. I went to timeanddate.com and I figured out the times. 8:02, 8:17, 8:29, 8:32, 8:20. You guys can read. Look at all those numbers. Blah, 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 blah. That is not the answer to my project. That is not my conclusion at all. This is data that I would need to put in some kind of form that makes sense. Like, can you study the numbers and then put them in a graph or a chart that somehow shows a pattern? What might that chart look like? Is that a pie chart? Is that a line graph? How could I show that change over time? Can I predict sunset times for next year if I find a pattern? Probably. If I did see a pattern, or if I did want to do something with that analemma, do I make a drawing of where it is every day? Can I use a sheet of paper and just put a dot where I see it every day? How would I measure where that dot is? Would I use a 360 degree along the horizon? Circles are 360 degrees all the way around. Would I just use the cardinal directions, north, south, east, west? Will it show a pattern? That's what you do with your data. And a really good analysis of data can make amazing charts and graphs that can take something really complicated and make it easy for even someone that doesn't know what they're talking about, understand it. That's the beauty of science. So are there any questions about this phenomenon, about not being able to play outside after supper anymore? Oh, by the way, I'm not here to just throw out answers at you necessarily. I'm wanting you to think about the questions that you have. Think about questions that you can maybe classify. Is it a pattern question? Is it an observation question? Is it an analysis question? And maybe the other panelists could share a couple of questions that you have right now, and maybe I could show a little bit of it without totally going into the answers.
I had a question, Peggy.
Sure.
Would it be the same if I lived in Australia?
Wow. That is a mind-blowing question. What an excellent field trip. Shall we go? Wouldn't that be fun? Go to Australia and do the same thing. Watch the sun every day and see if it does that same pattern. So does the sun rise and set in Australia? Would we even be able to do it there? Does the sun rise and set everywhere on the earth? It should. We should be able to do that. And I'm actually still sort of new at Stellarium, and there is a setting to go in and change your latitude, and it's a little above me right now. I haven't done it in a while, and I can't remember for sure how to do it. But that would be a fantastic project to do. And as a matter of fact, boys and girls, the Stellarium program that I'm using to show you these things, I want you to know that it's a free download. It's a really neat program. Planetarium presenters have been using it for years, so it's a free open source program. And you could actually come up with your own observation investigation, and maybe you could set yours for Australia and then compare the Australia and the one here in the Elgin area and compare the two and try to figure out why they look the same or why they look different. That would be excellent. So again, there's all these settings on the side, the sky and viewing options. There's a ton. It takes a little time to learn it, but you could move yourself to Australia and see if it does that same figure 8 pattern. Any other questions about it getting darker earlier?
Here's one, and I'm trying to, we may have to ask this person for a little bit more detail, but I think what they're trying to ask is, how does the time change in different areas? So if it's 6:55 here, that time in other places is what I think the student is asking.
Okay, so they're not asking about changing to daylight savings and then back in the
I
spring
don't
and
think so
fall.
because
It's more about, okay, so the fact is, wherever somebody's standing on Earth, the exact moment of noon theoretically is the exact moment that the sun is directly in the south. Now, this is a little bit past noon, so I could, this could show you this. So if we go back and help me line it up, does that look straight? Yeah, I'd say that's the very exact moment. But look, it's saying 11:45. Well, that's because we chunked out the Earth in these big, broad bands of time zones, because if somebody 10 feet away from me has noon 15 seconds after I do, and I say, meet me here at noon, I'm going to be 15 seconds early or late from that person. And yet I'm standing right next to them, and that doesn't make any sense. It doesn't make any sense for trains to have a schedule if everybody has their own time based on the exact moment the sun's going to be in south for them. So we kind of lump it all together in these bands we call time zones of thousands of miles across. And if you go a couple of thousand, if you go 1,000 miles to the west from where we live, you are definitely going to be in the mountain time zone. And then you're going to end up in the Pacific time zone if you go all the way to California. And that's because our Earth is round, and noon is at a different moment for different people on different sides of the Earth. It's different for people that are an inch apart, but instead we lump it together into these time zones that go by bigger chunks, just to make sense. Hopefully that addresses the question.
I think that does. So a question that's kind of an interesting one. It's a little bit different from this physical stuff, from the Earth and space science that we're talking about now. But the student is asking, is disappointment a phenomenon? And I think that it could be.
What is disappointment a phenomenon?
Yes. Is that a studyable phenomenon, disappointment?
Wow. Yeah. It's not, that wouldn't be, sometimes people would say that's not a hard science, but yeah, the science of studying people and the human brain and feelings and emotions, that would be very interesting. Like, why are some people disappointed at something, but a similar person, a similar layout in somebody else doesn't cause disappointment? And some people, when they're disappointed, act differently than other people when they're disappointed. So what drives that? So those are excellent questions that would go with that phenomenon.
It could be a perfect pattern also. If you wanted to take one topic of something and see if once you bring this topic up, are people consistently disappointed, or what are their feelings?
Yes. And what makes people get disappointed? Like, are there triggers that make people disappointed that you can remove, and then people won't be so disappointed? Interesting. Yeah, I think that's an excellent idea. The social sciences definitely have scientific investigations that go on.
It's just so fun that patterns and structure and function, you just look at those things and you can look at so many scientific phenomenons just understanding those two principles. It's kind of cool.
Yep. And they cover all sorts of sciences, not just microbiology and cells and astronomy, but you could go to all of them. Geology, you could go into physics, you could go into other life sciences, and there's patterns in every single one of them. And if you find a pattern that you're interested, you could make a project out of it. If it's a pattern that you've noticed in a phenomenon, it's worthy of a project. Some great questions. And I really just want you to be thinking about questions. Mm-hmm. And I noticed that it's 7:00, and this is technically the time that we're going to be done. But how about if I go to today, November 11th, and we watch the sunset and we go through the evening sky tonight, kind of give you some pointers for what's coming up, what's going on. And then we'll do a quick Q&A at the end as well. And we're right at about noon on November 11th today. And this is exactly what happened. The sun came up over here in the east. It's rising across the sky. It's actually moving right now. But in real time, the spin of the earth that takes 24 hours, it's hard to notice the sun moving. So let me fast forward it a little bit. And hey, notice that the moon and the sun seem to be moving kind of at the same speed. They're definitely both moving from the east to the west. And we're already at 12:30 now, 1 o'clock, 1:30, 2 o'clock, and down goes the sun in the southwest. 4:38, that's about the time of sunset. And it's definitely getting dark out. And in the west, we have Jupiter. Jupiter is our most obvious object in the western sky as it got dark out tonight. At 4:40, when we had that beautiful sunset, is when it was noticeable. We will start seeing stars any minute, and I'm fast forwarding time, and I want you to know it's very common, boys and girls, for there to be satellites every single night. As it gets dark out, there's satellites right now going past in the sky. You can't see them though, because it's not dark enough in the background yet. But once we get to dark, you're going to see lots and lots of satellites zipping by, and you'll see that the stars are coming out. So I'm going to fast forward. We're at 10 to 5 right now. Now I can see Saturn. Jupiter and Saturn are both in the southwest as soon as it gets dark out tonight. It is dark enough to see stars by 5 o'clock. Still a little pinkish orange though in the horizon. So we're done at 7. We'll go to 7 o'clock because that's about the time that it is right now. And I'll slow down. These are the stars that are up tonight in the southwest sky. Hey, I'm still noticing a movement. All these stars look like they're heading down in the southwest. They seem to be following that same path that the sun and the moon took across the sky. Hey, the stars, the sun, the moon, they all follow the same path. Is that a pattern? Is there something causing that pattern in astronomy that I could study, that I could do an observational investigation on? Something that I could look at to figure out why it's happening? Of course I could. And that could be another STEM Expo project for you. So here we are at 6:45, close to 7 o'clock. I've seen a lot of satellites go by. Did you see any constellations that you know? Do you see anything that you recognize? I'm going to pause right here. These 3 that make up a triangle, that's called the summer triangle. I know it's November, but you can see the summer triangle in the west all the way into December. Definitely notice Jupiter and Saturn. If I drew in the lines for some constellations, they kind of look like that. Deneb is the Northern Cross. This time of year, she's dive bombing straight towards the horizon. She's sometimes called the marker of the Milky Way. And I don't know if you can notice this hazy line going through here. That is our Milky Way. That's our eyes looking through billions and billions of stars out in that direction. And they're so thick that they make the entire background glow. So if I take those off and add in the drawings and the names, those are the Greek astronomical characters that we see in the evening sky in November in the southwest. Nothing super, super bright other than that summer triangle. If I look up a little bit, so it gets a little distorted, but pretend like you're turning your head up in the sky. I think the neatest one to look at is Pegasus, right here, the Great Square of Pegasus. It's not too far from Mars. That's Mars right there. And if let's go to the south a little bit. If I turn my whole body, I'm going to spin to the left. And if we're facing southwest, I can still see Jupiter and Saturn. Mars is well up above the horizon. And above Mars is where we have that Great Square of Pegasus. It's a pretty big square in the sky. It's actually a pretty perfect square outside, and there's not a lot of those in the sky. And the neat thing is, this is Andromeda right here, going off that upper left-hand star in that box. And right here, that little fuzzy spot, that's the Andromeda Galaxy. It's not a star, it's a fuzzy spot. And depending on your screen, I know it might be a little bit hard to see, but that is the Andromeda Galaxy. It's the only galaxy that we can see naked eye in the northern hemisphere in a dark sky. You have to have a nice dark sky to be able to see it. Mars is in Pisces the fish tonight. There's the drawings. And there's all the names. Now, if we go off towards the east a little bit, or you know what, I'm going to go a little bit later right now. And if I fast forward a little bit, you can kind of tell as we're facing south, everything's rising in the east and setting in the west. And later, so here it is 9:30, 10 o'clock, 10:30. At 11 o'clock tonight, we start to see constellations that we see normally in the evening in December, in the winter. And what we've got is Betelgeuse, Orion the Hunter with bright Betelgeuse in the shoulder. Here's Sirius, the dog star coming up. And right here is Taurus the Bull. It's a letter V-shaped constellation. And in this area, this is the radiant for a meteor shower the next couple of nights. It's not prolific, which means the North Taurid meteor shower that's coming up the next couple of nights, best to look after 11. I know it's not okay on a school night, but you might catch some this weekend, Friday night. There's not a lot of them. You can't go outside and expect to see a shooting star or meteor like every minute. But if you do see one of these, they typically are very big and relatively slow as they cross the sky. So they're good ones to spot, the North Taurids, and that is the next couple of days. Those are meteors, extra meteors, because we're going through the path of a comet, where a comet went through and it left behind debris. So at 11 o'clock tonight in the east, I've got Gemini the twins. I can see those two right there. Right there's Gemini the twins, Orion, Taurus. And off to the left, I see the Big Dipper. That's because the Big Dipper is what we'll see in the north. And I'm going to end with one last pattern here. Here we are in the north. Big Dipper and Little Dipper are there. The Big Dipper is really low on the horizon this time of year. And right now the handle's actually sticking down below the horizon in this diagram. Oops. So right here is the Big Dipper. Part of the Big Bear. There's the mama bear's snout. There's her front legs and her back legs. And the last two stars in the saucepan aim right here to this star. It's not super bright. It's like the 49th brightest star in the sky. So I don't know why anybody would ever call this star right here like, woo, super bright, because it's not. That is the North Star. That's Polaris. It's always in the north because that's the star that the North Pole of the Earth points to as we spin. And I already said too much. I said I wasn't going to say too much. I want you to notice something, though. I'm going to fast forward time, which I can do with this software. I want you to stare at the North Star right there in the Little Dipper, right there. Just stare at the North Star. It's 11 p.m. now. Here's 11:30. Here's midnight. Here's 1 in the morning. Do you notice anything special about that star and what happens in the northern sky all night long? I mean, if we head back to the south and compare what's happening over here, same thing. So now we're looking in the south. In the south, everything rises in the east and sets in the west. But in the north, we had everything doing something very, very different. A pattern like this, a circular pattern. Could you do an observational project on the North Star in that? Of course you could. You could definitely find a pattern there. So now we are tomorrow morning at 9:52. Of course, when you go outside, you aren't going to see all those lines, but they're there. I mean, we just can't see them because the sun is so bright. And we are at a waning gibbous moon, or a waning crescent moon this week, which means every day it's getting closer to the sun, getting closer and closer to a new moon. So this moon is impossible to see in the daytime sky when it's up, and it's impossible to see on the nighttime because it's on the daytime side. So we do not have a good couple of days for observing the moon. Okay, so are there any questions especially maybe a question about patterns or I'll answer questions about tonight's sky too.
There's it's not necessarily a question as more of a observation. If on January 1st, 1776.
Hold on. You know what? That is excellent, because sometimes we get stuck making science projects because we keep making a statement that we already know about, and we try to make it an investigation, but we already know the answer. And you can't make a phenomenon, a problem, without having some kind of a question. So not that statements are bad, but when we start our projects, we got to make sure we start with a problem or some kind of a question with our phenomenon. But yeah, so sounds like you've got a history thing going on.
They were just wondering if, January 1st, 1776 sunset might be the same as January 1st, 2020.
Reasonable question. So,
Mhm.
okay, January 1st, let's see, I've never gone that far. Can I just enter? Okay, what year? 1776 you wanted to see? 1776.
That's right. That's correct.
And we'll go to January 1st. And what were we looking at? Sunrise, sunset? What did we want to see?
Set. And the student was also using Stellarium to do it.
Oh, interesting. So,
so there we go. There's a project.
yeah. So yeah, it's fun to play around with. And there can be questions that come up, and that should be a light bulb that says, oh, hey, I could do a project on that. And you absolutely could. And it's fun. So sunset on January 1st, 1776, you can totally do it. And it looks like it's at around 4:22 on January 1st, 1776. So could you change the date and check different dates? Absolutely. And you probably would find some kind of a pattern or an investigation built in there.
We've got a question about, can patterns be how something reacts?
Could a pattern be how something reacts? Well, yeah, but but maybe not. Depends on how you mean. But if you keep repeating something and you keep getting the same reaction, then that's definitely a pattern. And if it keeps happening every single time, that's a very reliable pattern. So yeah, if a reaction repeats, then yes, I would call it a pattern.
And it might be interesting because I'm thinking about, when I was growing up, my sister would scare me for fun, right? But if she kept scaring me over and over again, I started expecting it and I stopped reacting.
Oh, interesting.
So, I mean, that might be another investigation someone could do.
Yeah, that sounds like a social science. Mm-hmm.
And I'm also thinking about, when you, I'm thinking about migration. It's the time of year when we've got salmon are going to start migrating, right? And so when salmon, salmon are migrating to warmer waters where they're going to be able to survive the cold. And so when they do that, then predators are going to go into those same parts of the waters. So that might be another pattern that is sort of a reaction.
Interesting, because I would think there would be a reaction to animals that might like to eat salmon, have this reaction because of this pattern that they head towards the river at a certain time every year because they know there's going to be extra salmon there.
So maybe some animals react to each other and follow the same patterns. They have patterns that match.
Yeah, that could be interesting.
I think we've proposed a lot of questions, and I really appreciate everybody's attention that we went a little bit long, but we knew that you'd like the stellarium to see what was up in the sky. And And I still see questions coming in. So we know that people might have to drop off. We can hang around. I know I can hang around and answer questions if people have them. And otherwise, that was really fun. And we hope you come back next week. Could a pattern be something like if more salt is placed, something happens? So patterns can be anything, really. Patterns can be anything. And
Well, but think of it as, can you use it to make a prediction?
right.
Because if it's something that repeats and it's predictable, then it's a pattern. But if you place more salt and you get a different reaction, that's not a pattern. That's a different reaction because you added more salt. So be careful in trying to make a pattern when it's not there. But a pattern is repeated. And because of that pattern, you would be able to predict. Like, I'm going to predict that there's an eclipse that's going to happen on April 8th, 2024, because we've studied the movement of the sun and the earth and the moon. And those three, there's going to be a convergence, a lineup of them. So how would I know if that's a pattern? Well, I'm going to have to wait till April 8th, 2024 to know for sure, because I'll have to find out if my prediction really was correct. And then I can really say that was definitely a pattern that I could use to predict.
Well, this was fun. We've no other questions coming in, just lots of compliments. Lots of people enjoyed today.
Good.
And I did too. That was fun.
Yeah, we're going to have more fun next week too.
Yes.
I can't wait to see the dinosaurs.
Me neither. Come back next week for paleontologists and dinosaurs and more fun from your science staff. So we have Frankie Valencia, who is a science coach. We have the Sarah Rodriguez Horn, incomparable Sarah Rodriguez Horn, who is our office guru, keeps us all in check. We have Vince Fosco, another science coach, instructional coach, and then Peggy Hernandez, who runs our planetarium. And then we have our special guest, Erica Sotek, who taught you about structure and function. So we hope you guys all come back next week. Tell your friends. Tell your friends.
Yep, it's open to anyone that logs into the webinar.
Yep. And it'll be the same link, by the way. It'll be the same link for next year or next week. Oh, I know what I'd like to do. Yeah, you can start your project anytime.
Definitely, especially if you're going to do a sky observing investigation, you would want to start as early as possible and just collect your data.
Should they could email either myself or Mrs. Hernandez, and we can get you the download for that program. We can get you the link so that you can download Stellarium. Can we use a trifle board? Oh my gosh, that's such a great question. I'm so glad you asked that.
Oh yeah.
Yeah, here's the good and the bad. We're going to be able to do the STEM Expo, but we're going to have to do it virtually. And so we're going to actually do this via webinar. And instead of a trifold board, we're going to be giving you a template for Google Slides. So you're going to build your presentation out in Google Slides. Then prior to March 20th, you can work with a team if you'd like, you will select a 30-minute time slot. You'll come into webinar. We will promote you to panelist. You will share your screen just like we did. You will do your presentation, and wonderful people like Erica Zotek are going to ask you questions about your project. And the great thing is that you're going to be able to give this link to anybody. You can give this link to your grandma, maybe if she lives in another country or another state. You can give this to your friends. You can give this to your class. You'll be able to invite anybody you want to come see your presentation or not. Maybe you're shy. I don't know. But it'll be here, and that's how we're going to do it. So great question. No trifolds this year. It's going to be all through slide decks. When you register, we'll send you a packet of information, and it will include the template for your slides.
So the slide deck is going to be the substitute for your trifold. You can put pictures on it. You can take pictures on your phone and just upload it onto your slide deck. You could make a graph on PowerPoint and you could put it in a slide deck. You could do it right on Google if you want, if you know how to use those features in Google. So you're just going to put everything on your slide deck and just click through it while you talk as a panelist, just like we did today. So great question.
do exactly what Mrs. Zotek did, and you could film it. You could film the whole thing. And let's say that you don't want to do this presentation live, like it just makes you a little bit nervous to do this on webinar. You could send it to us and we would still have people look at it and give you comments and ask you questions back and forth, but you don't have to do this format if you're a little bit shy. Or you could film the whole thing just like she did and then be available for question and answer just like she did.
Yep.
You will be able to register for the STEM Expo on starting in December. And if you've registered on our Google Sheet, so if you have the link for today, chances are somebody in your household registered. We will be sending that Google form registration link to all of you who have registered to be in the webinar, but we'll also be sending it out to teachers. We'll have it on the website. It'll go out through the different U46 channels. So, or just email me anytime. I'll put my email in here. And you can also email me in December and I will send you the link to register. And we'll be asking you all these questions. Like, do you want to do this live? Would you rather just,
So no, today was not registering for the STEM Expo, but by registering for today, you got on the email list that when that Google form is ready for you to fill out, you're going to get an email that says, hey, register for the STEM Expo today. And that'll be in December.
we just wanted to send you information about our 4 informational sessions so you can get to talk to all these interesting scientists and get some ideas. All right. If there are no more questions, I'm going to go ahead and end the session. It was wonderful. Thanks to all my my science staff, by the way. You guys are awesome and amazing, and I appreciate you. And thanks again, Erica. We appreciate you too.
Yeah, thanks for coming, Erica. It was fun.
Thank you. I enjoyed it.
I'm so glad you love this. I'm so glad you guys all loved it. Come back next week. It's going to be really fun.